All right, everyone, we're going to go ahead and get started with our panel discussion. If people can grab their seats. All right.
Yes, sir.
All right. So welcome to our panel discussion portion of the day. We're going to be talking about the value of collaborative data and resource sharing to advance ALS research. The discussion will be moderated by Danielle Boyce and will feature Fernando Vieira, Tonya Gilbert, Irina Antonijevic, and Dyson.
Okay, can you hear me? Am I on? Okay. So hi, thanks everyone for staying for this great panel discussion. I've been really excited in talking to all the panelists earlier about about what you're about to hear. I'd like to start just with everyone introducing themselves and their connection to the panel and the topics that we're talking about today. Can we start with Tonya, please?
Sure. Hi, everyone. My name is Tonya Gilbert, and I'm the Executive Director of Translational Research at Eikonizo Therapeutics. Eikonizo is a biotech company located here in Cambridge, Massachusetts, and we are developing disease-modifying small molecule therapeutics that target HDAC6 to improve the lives of people impacted by neurodegenerative diseases as well as cardiorenal diseases. Eikonizo has had the pleasure of collaborating with ALS TDI on preclinical research for over 5 years now. I can't believe it, and it's a real honor to be here today. Thank you so much.
Can we? Thank you. Took me a minute to figure that out. For most of you know who I am now, but for folks who are maybe arrived late or tuning in online now, I'm Fernando Vieira. I'm the Chief Science Officer and CEO at the ALS Therapy Development Institute, where we're a nonprofit research institute focused on finding treatments for ALS.
Hello, yeah, it's on. I'm Irina Antonijevic. I'm the Chief Medical Officer at Trace Neuroscience. Fernando very kindly mentioned us before. We are a relatively early stage startup. We have been really sort of in existence for a little bit more than a year. We are developing an antisense oligonucleotide targeting a specific genetic modifier that has been associated with ALS and with faster progression in ALS. It's called UNC13A. And we hope to be in the clinic in the next year, and we think that this has a good chance, because it's genetically validated target, to really make an impact on people living with ALS and also people living with FTD and potentially even people living with some forms of Alzheimer's disease. Thank you very much for inviting us.
My name's Dyson. I'm a patient with ALS. I was diagnosed about 2.5 years ago. I actually have the UNC13A variant, so I'm going to talk to you later. But I'm also responsible for the Longitude Prize, which you'll hear about later on today, which has involved pulling together large data sets from around the world, including in partnership with ALS TDI and others, so that we can create a competitive environment for examination and identification of new targets.
Thank you so much. So I have a question that I'm going to start. I think I'm going to ask you, Irina, if that's okay, and then everybody can jump in and add to your response. So can you share an example of where data or resource sharing has genuinely changed the trajectory of ALS research or care?
So you mean very generally,
yes,
not specifically?
Very generally.
I would say the foundation of Trace is based on genetic research. And the problem with genetic research is that sometimes there is a small effect, but it's still a relevant effect. But in order to get to that, we need to have many, many people from whom we have the genetic data. And so for that, we need really collaborations across, multiple organizations, multiple countries, because only that's how we can discover some of these genetic targets. So this, I think, particularly in ALS, has made a huge impact and is continuing to make an impact. We are almost every week learning about some new genetic, they're not necessarily mutations, they're just genetic variants that have been associated with ALS. But maybe just if I can add to that, so when this was first identified for UNC13A specifically, it was, and for C9 at the time, it was 2009. But it's only in the last few years that we have understood how UNC13A actually contributes to the disease. So the genetics alone is not enough. Then we had more collaborations, and one, I think, something very dear to me is the post-mortem tissue collection, because it's how we then identify how certain genetic factors might actually change the disease pathology. So I just want to say, anyone who has contributed to the tissue banks, this is really an incredible resource, and thank you for doing the research, and thank you for everyone who contributed to that.
Thank you. Does anyone else have anything to add to that question? Any examples where data or resource sharing has changed the trajectory of ALS research or care?
Another example that comes to mind for me is the platform trials that have been developed in ALS. One notable example of this is the Healy ALS platform trial. So this is a collaborative clinical trial that tests multiple drug regimens at the same time. And to do this, they use a shared protocol, a shared infrastructure of clinical trial sites, and critically, a shared placebo group. So this really maximizes efficiency across the different drug regimens, and it allows more participants to be on active drug. Through this, you are able to decrease the time it takes to complete a clinical trial by around 50%. You can reduce costs, and you can also increase participation. Microphone tag today. This has allowed the Healy trial to complete the testing of 7 drug regimens since 2020, which I think is really incredible. And this is paving the way for other platform trials and approaches. So, for example, there is the Experts ALS program that's coming out of the University of Oxford. And this is a pretrial program that tests multiple drug regimens on the basis of their impact on NFL. So candidates that lower NFL are then going to be prioritized for larger clinical trials. And I think that platform trials is really the way of the future for doing ALS clinical research.
Yeah, I might just add about, because the Healy platform is obviously remarkable and Experts is quite new. I think one of the advantages of Experts is it's quick. It's 6 months. There's no placebo. They're just looking at neurofilament drop-off. And it is way, way cheaper. So if you're at a point where you've got a molecule, but it's struggling to raise vast amounts of money, this is a potentially really good option for you, I think. And yeah, it's run out of the UK.
Yeah, a few different points on this. I guess focused on ALS TDI just for a moment. It's been really, really incredible to see the value that's come out of the data sharing from the ARC data commons, particularly around actigraphy, so the accelerometer measures. It seems like getting that into the hands of other researchers and other experts in the space has really accelerated the value creation from that, and we're starting to see that taking root with different organizations using those as they're designing their clinical trials. I also want to shout out Answer ALS, particularly for their cell biorepository. They really collected a really robust set of induced pluripotent stem cells from their participants in that study over many years, and we're able to share those with a lot of academic investigators over, you know, a half decade or so. And we've seen a lot of different publications and a lot of really interesting findings and therapeutic targets emerge from that. So really, I think the overall field in general over the past decade has started to break down those silos that we've heard about and really tried to be open and again, kind of lift all boats for the benefit of the patient community.
So, Fernando, you touched on this, that in early stage biology, duplication is common across labs. And you mentioned Answer ALS is a good example, and our obviously the ARC data set and the tools available through ARC as being an example of that kind of infrastructure that can reduce that duplication. But what can you think of, any of you, this is to the whole panel, can you think of any other data sets or infrastructure or resources that would help to foster, you know, with that not having to reinvent the wheel? And if so, what would it take? Importantly, what is it going to take to have more of those types of opportunities and resources available?
So is the focus on sort of reducing duplicative effort or on the value of parallel efforts, right? And happy to speak to both. Yeah. So I think it's sort of a project by project type consideration. A lot of times when two groups are doing the same thing at the same time, everything in science requires replication. You need to see if it happens again and again and again in order to really trust it. And if you're doing everything in series, one, then after the other, then after the other, then by definition, that's going to take longer. Whereas if a couple of different groups are actually pursuing the same idea at the same time in slightly different ways, that can actually often be really valuable. Oftentimes you'll see pairs of papers from two different groups published at the same time, where it's really a stronger story because there's a replication happening there. So there's value in the much maligned duplicative effort. But at the same time, it's really important for us as organizations in this entire ecosystem to kind of look and see how can we be complementary. And the key piece to that is in communicating, right? In understanding and knowing what other stakeholders are doing, not necessarily saying, okay, well, you're doing that, so we're going to stop. It's more, oh, you're doing that this way. How can what we're doing complement that? And how can we be learning from each other in real time rather than waiting 4 years to figure out what happened and then see, oh, we should have been talking. And so I guess those are my top level responses to that.
I would just like to add something to that, because I do actually think the replication, as Fernando just said, is very important. But I was part of some roundtables, I think by David Taylor also, and trying sort of to set certain standards or certain protocols, how, let's say, people do some cellular work and they want to interrogate how certain drugs, new drugs maybe affect certain downstream effects at a cell model. And so in order, like the platform trials in the clinic, if we would have sort of standardized protocols where we could actually as a community compare drug A, drug B, drug C, and really sort of agree that we move forward what looks most promising rather than everyone developing this in their own lab with slightly different protocols. I mean, I think it's not an easy task, but I think if we can at least start by sharing protocols and maybe trying to get enough buy-in to have standardized protocols.
Yeah, I really agree with what you're saying. I think in early stage biology, we do have the opportunity to borrow the principles from things like platform trials, because in any preclinical research study, you always need certain control groups there, or else you're not going to have a valid study. And oftentimes you can kind of run into a bandwidth issue because you can only do so much at the same time. So I like the idea of, you know, the ability to find like-minded people who are doing similar things so you can share those control groups. You don't always need to have them duplicated. And also we can think about doing things like sharing sample testing. So a lot of times a particular technology, it might be out of reach for an individual group because honestly the costs can be very high. But some of these technologies have economies of scale. So if you're able to group samples from multiple groups together, you can really lower things like sample minimums, and you have the ability to do more with less. And to make this happen, we need ways to find each other. And I think today is a great example of that, bringing together researchers with like-minded views on collaboration. And also we can use things like simple message forums, just for special interest groups. But I'd love to hear other ideas as well for how we can really bring together people that would be willing to go in on studies together.
Yeah, and I think what we've been touching on a little bit here is the pre-competitive collaboration. And I personally have been involved with a number of these pre-competitive projects in the rare disease space. In particular, there's one that comes to mind. There's a mitochondrial disease task force at Critical Path Institute that I'm a member of, and that brings together people from industry, patient advocacy groups, data holders, regulators, and it helps, for example, finding a good outcome measure, putting everybody into the room, and also it reduces duplicative efforts in the sense that maybe a patient group thinks they need to create their own natural history study, but one already exists that they can join. So can you think of other examples from either oncology or rare disease that might be models that we could borrow from here?
So I've also worked with the CPAS Institute in the past and many other rare diseases. And I know that it exists for ALS, but it doesn't seem to play such a prominent role as some other diseases. I'm relatively new to the ALS community, so I don't know exactly why, but I think if we could try to bring different organizations even under a same umbrella, so that we, like endpoint development, nobody can develop an endpoint just by themselves. Yeah? We need the collaboration, we need the replication, we need regulatory buy-in, we need the technology to sort of hold up. So I think this would be great if it doesn't have to be CPAS, but whatever it is, sort of to bring together these various resources.
I was going to say, I mean, my observation as a patient is, and I think you alluded to this earlier on, is it all just takes far too long. And so we need to speed things up. And I think there's a heavy suspicion amongst the research community that successful treatments are going to be multimodal. And as we're starting to understand that particular approaches may tackle different modalities, I think we should be considering, particularly when we're looking in, iPSC lines or maybe in animal models, looking at those combination approaches. And I think the research community often doesn't like that because it messes the data up. It's not clear. But I think there's also a potential risk, which is if you're just aiming a drug at one modality and there's like 5 or 6 things that are going wrong, that drug may be working, but it's only working on that one modality, and so you're not picking it up properly. Anyway, I suspect there are lessons to be learned from things like the way AIDS and HIV was looked at in the 80s, but also the way that we pulled our finger out on COVID by bringing companies, regulators, and universities together and move things much, much more quickly.
I wanted to speak to, Irina, your point on the sort of assemblage of a consortium around clinical endpoints, and CPATH in particular. I think you're right. It hasn't been as prominent because it is new. And I think we owe its entry into the space in large part to Act for ALS, which did ultimately end up funding AMP-ALS, which is this private-public partnership, which those funds are potentially at risk, but one of the outcomes of that has actually been an effort to certainly look at the overall omics of ALS, so sort of the underlying biology, but also to look at the endpoint measures and what's going to be meaningful for people with ALS. And so we've been fortunate to really connect. It's really been in the last 6 months that a more robust working group has emerged looking at these digital outcomes and endpoints for ALS. So we hope to gain more traction, maybe in Q1 of 2026, to maybe get a letter of intent in front of the FDA, at least around accelerometry. And then, Tris, on your point, I do agree that we need to find ways to tackle these more complex biologies, right? With looking at multiple modalities at once. And maybe this is a domain where, and I'm kind of like lobbing a softball to you, where artificial intelligence could be really key to parsing data in ways that have been messy and complicated and unwieldy for those in the lab trying to understand, and just get your thoughts on that.
No, I agree. I mean, the stratification of patients and understanding of different modalities is certainly something that I think some of these new models will be able to help with these sort of, particularly as the data gets bigger and bigger and more easier to examine. So, yeah, but I think we also need to be prepared for when that information starts to come through, how we're then translating that into the wet lab.
Yeah, maybe just to add on a little bit to what Fernando was mentioning about omics data sets. I think there is additional opportunity to continue building out biomarker consortia. Today we heard wonderful updates on the ARC study, also the NeuroMind platform. There's also the Target ALS natural history study. And these efforts are critical for using, for example, proteomics to identify novel biomarkers for ALS, because you're able to look in a large number of participants over a really long time frame. And so you can learn how the trajectory of these markers is really fluctuating over time. I think it would be interesting to start adding in data from industry-sponsored trials so that we can learn about how biomarkers perform in an interventional setting. It's critical for sponsors to be able to understand what they can expect from biomarkers in a relatively short time frame with a relatively limited amount of people. So these kinds of data would really help us select the best suite of biomarkers that has a chance for showing meaningful outcomes.
So before I open it up to questions, I think we're going to do one more question. And most importantly, none of this would be possible without people with ALS or at risk of developing ALS donating their data and participating in these studies. And so what can we tell people in the community, people with ALS, to help them feel confident that contributing their data is worthwhile, that it's respected, that they're protected, their data are protected? How do we communicate this need and these? I mean, obviously there's a sense of urgency that everyone knows, but it takes time to contribute data, and people are worried about how their data are protected, and they're also worried that it won't be shared. So what can we tell people participating in these studies that it's worthwhile?
I'm happy to go first. Just from the perspective of the ARC study, it's when we set up the ARC study, and continuously as we continue to run the ARC study, we do have to present our protocol to an institutional review board, which is external to the organization, which really evaluates the ways that we are protecting the data that participants are contributing. We are incredibly indebted to everybody. Like people, one of the ways that it becomes really, really tangible to us is when we listen to the voice recordings. It is an incredibly real and visceral experience to hear a person with ALS contributing their voice sample, which is difficult, particularly if they have bulbar symptoms. And so it's incredibly meaningful, and we work hard to honor that. As far as a sense that it's valuable, right? And I think we're seeing the biggest returns from the sharing of it, right? The making it available to other individual investigators. We find it's a really multimodal study where lots of different data types that we're collecting. And so there are experts in actigraphy, but there are also experts in genomics, and there are experts in proteomics. And so these different eyes on these different lanes of the data, it's incredibly important because we can't have all of them, right? I just named a bunch of different specialists that we didn't have to hire. Right, who are analyzing the data. And then imagining taking that data set and then making it even more available, right, through other channels, through sharing with NeuroMind, through sharing with Longitude Prize, et cetera. You just increase and amplify the reach of something that somebody in a house was spending a bunch of time contributing. I hope people understand the power and the lasting power. It's not just that day. That gets used over and over and analyzed over and over. It lives forever. And so thank you for that.
I mean, I'd say my main concern is when data is not being shared. Because, I mean, you spoke about it very powerfully. I think in the United States, you've got a high degree of openness around sharing data, which is really very positive. But the data is also scattered around a lot more, so there's a lot more need to gather it together, which is happening through, you mentioned Answer, I don't know if John Landers is here, but that is happening. But in parts of Europe and other areas of the world, people are sitting on these data sets, and they're not sharing, and they're often not sharing with private companies, where sometimes they're only sharing with research institutions. And they're doing this apparently in the name of patients, because they claim not to have had patient consent. And I think this is outrageous. I think they're holding back potential development of important studies that will lead eventually to treatments. And I think there's a confusion between what's regarded as personal information and personal data. I don't want people sharing my bank account information or any of that kind of stuff. But you can have my MRI scans, and you can share it with whoever the hell you want, quite honestly. I find it very frustrating when this is used as an excuse not to do so. However, when data is being shared, I think patients, as much as is possible, it would be useful if we were kept informed about developments and what's happening, and particularly if you're on a study, you should be able to get some access to information rather than waiting to the end of the study to find out what's happening, I think.
Yeah, and just to tie on to that, I think that it's really important that we share and publish data, even if it's inconclusive, even if it's negative and it didn't move the needle the way we hoped that it would. This is crucial so that people aren't going down the same unfruitful paths. So I think that that needs to be an expectation when we're collecting this data and people are donating their very precious time, that no matter the outcome, we're going to put it out there so that people can learn from it.
Yeah, and I just wanted to add that we have been thinking at Trace Neuroscience how to make, for instance, posters or presentations that we give more accessible. And so we were just sort of discussing just sort of the technical background to that, so that we could have this on our website, so that everybody who goes to our website can see all the posters we have presented, all the data, including the data that came from, like, for instance, TDI. And we also hoped to, so when we launch our clinical trial, hopefully next year, we wanted to have a site on our website where we report on the progress of our clinical trial, and we had also some council discussions, so with patient and caregiver councils, and they said, whenever you plan to update the investors or the market, please update us first. And so we took this very seriously to say, okay, we have these sort of calls where we say anybody, not only people who are part of the trial, who is interested in that, this is when we will share this, and then we will also post it on our website. So this is at least the plan, because we recognize that without the data that we get, we would not be able to design our trial in the way we currently have. So thank you.
One plug that I'll put out there is for DataCite and other communities that allow us to put DOIs and metadata on things like presentations and posters and things like that. We're a member of DataCite at ALS TDI, and so when we have a GitHub or something like that, we can put it in and it makes it findable. It has those characteristics that we're looking for in shareable data. The other thought that I had is that I've heard a lot from the community is the importance of setting up your study consent form in such a way that we can share the data after the fact. Because a lot of times people say, I can't share the data because they didn't consent for that, or we only said we were going to share with one group and not the other. And I feel like that might be a role for some of these pre-competitive consortia, at least the ones that I've been on. It has been really useful. A good example is the word de-identified, and what does that mean? When somebody says, we're show your de-identified data, but maybe their institution defines that differently than the data, so they can't share dates, for example, anymore, and that makes it less useful. So I wonder if you have any thoughts, anyone has any thoughts on that, like just the administrative. I know you were talking about protocols, but administrative things that could be agreed upon to facilitate sharing.
I mean, I personally think that the changing the informed consent form is probably absolutely key. And then, I mean, to your point, de-identifying, anonymizing the data. I mean, we just got some data after 12 months of back and forth negotiations due to the European strict regulations of getting access to data. So that the organization that had the data was willing, we were willing to pay for the data, the informed was covering it, but it was just the legal stuff, So it was really, really painful. But I think the informed consent, if this can be also not specifying with whom exactly to share, just specifying that no personal, no identifiable data will be shared, I think this would ease some of it.
And no attempt will be made to re-identify that kind of thing. Go ahead, Tristan.
This is a little bit different, but we were talking about this earlier, and I think institutions, are going to struggle to share data. They need to have legal data sharing agreements and all that sort of stuff. With pharma, biotech, there's obviously IP concerns and all that sort of stuff. So there's always going to be some problem with data sharing. But I think, I mean, you were talking about this. A potential solution could be a federated learning model where you're using generative AI to generate synthetic data off different data sets to create one large data set. And I think that's a very exciting avenue to pursue in the future, I think. But you were suggesting it, so maybe.
No, I'm glad you brought that up. You reminded me. Our project where we harmonize data with Answer ALS and Critical Path Institute was using the OMUP Common Data Model, Observational Medical Outcomes Partnership Common Data Model. And basically, in a nutshell, what that allows us to do is everybody organizes their data in the same tables. It calls everything the same concepts and the same field names. And then we can write a script that you run on your data, and we can aggregate the results, but we don't have to share the actual data. And it allows us to get around all of those issues with, I can't send you my data. That's okay. Just tell me how many people you have who meet this cohort definition, for example. And so I feel like that there's a lot of great work going on in Europe right now around that. Again, though, challenge is building infrastructure, extract, transforming, and loading, ETL your data, and all of that can be that requires some investment. But the tools are all open source and free to use, and there's a very robust community very interested in this. So I think that could be potentially an option too, especially for those where there's no way they're going to export their data. This could be run locally. So do we have some time for questions? Great. Thank you for our panel. This was great.
I'm sorry, it's Carmen Mikolajczyk again. I do have a burning question about AI, and maybe I'm speaking from my ignorance and my age, by the skepticism around. I hear you say that how useful this is to compile and to use all this data. And I have great confidence in your wonderful brains in organizing and reporting on that. But I'm also aware that how in space the slightest miscalculation mathematically can send things off into a wholly different orbit. So what are the guardrails and how can you be sure that what you're using, the way it's been quantified or reported by AI, is not misleading you?
Anyone have, I can, yeah, go ahead, Fernando, you can start.
I'll take a swing from my perspective anyway. So, and this is coming from the perspective of, like, we have a lab, right, where we get to test the ideas, right? So if ideas were to fall out of an AI algorithm of some kind, we would look for, does it hold up, right? Does it hold up experimentally in the lab? You don't move forward clinically, at least we wouldn't imagine moving forward clinically with an idea that was just conveyed through some type of algorithm, particularly one where you can't look under the hood and really understand what it said. But that's what you hope is, over time, you would iterate, right? So the outcome of the experiment that you ran in the lab would feed back in and hopefully improve the predictions over time. So when I think about artificial intelligence in the context of ALS, or indeed just drug discovery, drug development in general, I do imagine a future where it is just massive. It's the primary driver. When that future is, I don't know. How close we are to it, I don't know. And how much data we need to get there, I don't know. But I'm happy for us to play a part in helping that happen. So.
I can maybe just hopefully help to reassure you that we have just gone through an exercise where we had access to fully anonymized data from people with ALS. And we were asking a similar question. So we were looking at similar ways, but still requiring somewhat different data sets. And so we were looking at consistency of the data and also what we know what should happen. So, I mean, we did uncover that there were a couple of like smaller codes that had to be corrected because when we asked the question in two different ways, it wouldn't match, and we knew that they should match. And so I think there are really ways to reassure us that there isn't somewhere deep down an error in some of the coding and that we get erroneous data. So I think, but it's an important question, and it's important to do this, I think, also to exactly not go down a rabbit hole. Thank you.
I think it's definitely the right thing to be concerned about. I think if you run AI models at large data sets, they can come up with all sorts of things on their own. You know, maybe lots of people got big noses or something like that. So the AI has to be a partner, a lab partner to people with disease expertise and understanding of the disease biology. Otherwise, it's going to fire off all sorts of rubbish. That said, I do think it's a revolution that's starting to happen. If you think that, you know, we now have a treatment for SOD1 that is remarkable, well, that originates back in the 90s through data analysis, more conventional data analysis, computational approaches to genetic data back in the 90s where they discovered SOD1. This is that 1,000 times, and the problem with the disease that we've got is, although the knowledge of the biology is massively improved in the last 10, 15 years, largely due to increased research activity and research funding, it is extremely complicated, and that's why the potential of these AI models to get to grips with that complexity is phenomenal, I think.
Yeah, I think that question was really astute on a number of levels, and we have dabbled in like large language models in our work in extracting the data from the electronic health records, and there's an art and a science to it. I'm old school. I've been doing this for a really long time. I believe in, you know, working on a manual validation of models and things, a chart review, and then you use that to develop your code, and then from there you can feel more confident in the models. But the other thing, in addition to like algorithmic transparency, which is what you're, you know, part of what you're talking about is us sharing the code, is just the documentation on the data itself. So when you share data and you harmonize data, it has to be apples to apples, right? So we really need to do a better job. I've inherited so many data sets over the years where there's really no data dictionary. I really don't know what I'm looking at. How is it measured? Was it self-reported? Was it physician-reported? Was it verified? Was it, you know, all of these things. And so, again, part of this educational effort in the pre-competitive consortia could also be in just like this I call good data hygiene. You know, doing a good job documenting your code sharing it out on GitHub or some other data sharing repository, allowing people to test it and try it and validating it. It's an excellent question because, and it also needs to be interdisciplinary. There always needs to be a clinician or someone who knows what they're talking about on my team, not just a data engineer. It can't just be me, and that's why I often deflect questions about what I think about things clinically, because it's important that it's not what I think. It's I think about the data. I can have my own ideas, but we really have to rely on expertise. Great question.
Right, we have a couple quick questions before we have to break for lunch. So Maddie's got one on the mic over there.
I'm old and I'm new to the ALS community, but 25 years ago I was working on three-dimensional protein computations, and it was like, we're never gonna know what protein will look like if it's this sequence of 2,000 amino acids. And now they can do amazing things computationally. And they basically can tell you this is its conformation. And so I think maybe it'll be 10 years, but things like AI will be able to do amazing things as long as people are checking to make sure that what it says is what it is. And that's really what happened to the protein conformations. They come up with it, and it's like, well, it could be one of these 1 million conformations. And now it's this one conformation.
Thank you so much for that. That's ending things on a really positive note and a hopeful note. Thank you very much. And thanks to our panel. Thank you.
Danielle, we actually have two quick questions, if that's all right. We have one over here.
Oh, I thought we only had one more. Okay, sorry. That's okay.
Hi, how's it going? So I'm Kevin Zhu. I'm building a olfactory neuron platform as an accessible model for the CNS. And I guess my question's across the panel. So basically, what do you guys all see as the biggest blockers to improving our understanding of ALS with omics? Like, is it that we're not looking at the right biology? Do we lack the technology to measure the complex molecules in this heterogeneous disease? Or are we just looking at the wrong substrate because that's what we have access to in terms of, the wonderful resources like postmortem brain or biofluids, but fundamentally they're not, living neurons which are showcasing our disease?
I'm happy to take a first swing. You, as a biological entity, are a very, very, very complex being. Not just you, everybody here is, right? The data that we're capturing in any of these systems, whether it's induced pluripotent stem cell-derived motor neurons in a dish, single cell type, in a dish, in a slice of time, outside of the context of the complex milieu, a blood sample, a single or a collection of cell types, but outside of the complex milieu interfacing with, in the context of ALS, the brain and spinal cord. And these are all, slices of time. The data sets that we are collecting right now are massive, and we have a lot of data. And when you think about it in the context of everything that could be measured is a drop in the bucket. And my mentor, Al Gill, in the red jacket over there up front, has pointed out to me that we as scientists often like to line up the ducks that we have. Like, we take them and we line them up in a nice row. And we forget to acknowledge that there are 18,000 other ducks that we don't see. And so I think that is a very challenging and humbling reality that we confront, but we must work with what we have. And we have a lot more than we used to. And that's incredibly exciting to me. And to see that, the case that Tris referenced earlier of tofersen for SOD1 ALS, that knowing that critical piece of information for that subtype of ALS, which did kill my best friend, knowing that today, if he were diagnosed, he'd have hope to potentially get better,
means that the things that we are doing sometimes can be good enough and in time for people right now. So that's kind of like, I think of the big, daunting, scary reality, and I also think of the hopeful reality, and both are true at the same time.
And maybe I just add something from my perspective. So I'm a clinician, and I sometimes think that when we go back to examining a human being who comes to our hospital or our practice, and to do this really well and to do this in a really standardized way. I was talking to somebody who is by now very old, very wise, and he has worked in the ALS field for I don't know how many decades. And he worked together with a neuropathologist, and he's a clinician. And he actually said that, and I really believe that the heterogeneity that we always say is maybe less heterogeneous when we go really carefully back to analyze exactly the clinical presentation, exactly the evolution of the clinical presentation, and overlay it with the neuropathology of a very sort of, detailed neuropathology, that some of which we already have. And we can sort of maybe make better decisions on which groups of, people with a disease maybe show a more similar evolution of the disease, and then look at the biology. And I have to say this, I have been a clinician and a drug developer for many, many years, and I really think that some of the biology is strongest when we at least start with a smaller subgroup of very well phenotyped human beings, and then broaden from there. So we are trying to implement some of these very old-fashioned neurological examinations in our clinical trial to at least have this information. And again, this is where standardization becomes also very important. Thank you for your question.
Shout out to phenotyping. That's my job. Any more questions, Emily? Where's Emily? No, we're good? Okay, so I'm going to kick it back to Emily, who's going to tell us how we're going to eat.
All right.
Thank you again to our
Thank
panel.
you.
Thank you.